Okay, now we are starting the webinar. Myself, Dr. Harpreet Singh, the Alumni Coordinator, the School of Pharmaceutical Sciences, IFTM University, Muradabad. First of all, I would like to welcome and thanks Honorable Vice Chancellor, Professor M.P. Pandey sir, the respected Registrar, Professor Sanjeev Agarwal sir, and respected President, IFTM University Alumni Association, Professor Anush Srivastav sir, for giving us the opportunity to organize a webinar on role of clinical pharmacology in drug discovery and development that would be delivered by the eminent speaker, Dr. Yashpal Singh Chokar, who is currently working as Research Assistant Professor in Department of Pharmacy Practice and Science, University of Nebraska Medical Center, Omaha, United States of America. Welcome and thanks, Dr. Yashpal, for giving us your valuable time. Now, I would like to welcome the chairperson, Professor Navneet Verma sir, the convener, Professor Arun Kumar Mishra sir, and the organizing secretary, Professor Sushil Kumar sir. Now, I would like to invite Professor Arun Kumar Mishra sir, the Dean Student Welfare, who is also the alumnus of MPharm 2006-2008 batch, for his kind words. Over to you, Arun Mishra sir.
Thank you, Dr. Harpreet, for inviting me. Today, on the occasion of birthday of Rashtrapita Mahatma Gandhi, on behalf of Fraternity of Faculty of Pharmacy and IFTM University Alumni Association, I welcome you all in this webinar. I am thankful to our Honorable Vice Chancellor, Professor Mahendra Prasad Pandey sir, for motivating us to organize such type of webinar, which not only enhance our knowledge base, but the knowledge base of our student and also helps to develop a strong alumni connect. I welcome our registrar, Professor Sanjeev Agarwal sir, who is guiding force to uplift the scientific knowledge always. I welcome Dr. Navneet Verma, Dean Faculty of Pharmacy, and Dr. Sushil Kumar, Director School of Pharmaceutical Sciences, Head of Department Dr. Kavita Gehlot, alumni coordinators, faculty members, my dear postgraduate and undergraduate students. In today's webinar, we will learn the amalgamation of clinical pharmacology and its role with special reference to the drug design, discovery, and drug development. The speaker of today's webinar, Dr. Yashpal, is very renowned researcher, and along with good research profile, he is a very gentle, soft-hearted, and king of kind personality too. I know him personally and professionally both. Dr. Yashpal was my senior in my MPharm time and hostel mate in Dr. Asa Kaushik hostel. At that time, I have learned a lot from him, and even when he was doing PhD from CDRI, he was always keen to solve several pharmacokinetic and related problems of us. From bottom of my heart, I welcome you, sir. Apart from this all, I wish to convey one message from this platform to our students, that please listen to our speaker very carefully and be interactive in the stipulated time of the session so that query or the doubts may be addressed immediately. I hope this webinar will be helpful to uplift the knowledge of our student, and with these words, I welcome you all and thank you all. Thanks again. Now over to you, Harpreet.
Thanks, sir, for your Thanks, sir, for your valuable words. Now, I would like to invite the chairperson, Professor Navneet Verma sir, for his kind words. Navneet sir.
Thank you, Harpreet. Good morning to all of you. सबसे पहले मैं आज के एमिनेंट स्पीकर डॉक्टर यशपाल सिंह को बहुत बहुत धन्यवाद देना चाहता हूं कि उन्होंने अपने बिजी शेड्यूल में से ये समय निकाला कि वो इस वेबिनार को कंडक्ट कर सके यहाँ के स्टूडेंट्स के लिए, आईएफटीएम यूनिवर्सिटी के जो स्टूडेंट्स हैं, क्योंकि आने वाले समय में ये भी यहाँ के एल्यूमनाई होंगे और डॉक्टर यशपाल भी यहाँ के एल्यूमनाई हैं, तो इनको मैं बहुत बहुत धन्यवाद देना चाहता हूँ। रीजन मेन रीजन टाइम का बहुत बड़ा कंस्ट्रेंट था, क्योंकि जहाँ पे यशपाल हैं, डॉक्टर यशपाल इस टाइम, वहाँ के टाइमिंग में और यहाँ के टाइमिंग में लगभग 12 से 13 घंटे का डिफरेंस है। और यहाँ अगर सुबह के 10 बज रहे हैं तो वहाँ 11 बज रहे हैं, इस टाइम रात के 11 बज रहे हैं। तो उसको भी डॉक्टर यशपाल ने मैनेज किया और रात में उनके उनके टाइमिंग के हिसाब से रात में भी इस वेबिनार को करने के लिए वो एग्री हुए, उसके लिए थैंक यू वेरी मच। और मैं अगर किसी एल्मा मैटर की बात करूं तो हर स्टूडेंट के लिए, हर एलुमनाई के लिए अपना एल्मा मैटर यानी कि अपना इंस्टीट्यूट जहां से वो पढ़े होते हैं वो बहुत ही वैल्यू रखता है। और उसको आज डॉक्टर यशपाल ने साबित किया कि टाइमिंग को ना देखते हुए भी उन्होंने इस चीज के लिए कहा कि नहीं सर, मैं यहाँ के स्टूडेंट्स के लिए अपना जो भी एक लेक्चर है इस वेबिनार के थ्रू, जो आज का टॉपिक है डॉक्टर यशपाल का रोल ऑफ क्लिनिकल फार्माकोलॉजी इन ड्रग डिस्कवरी एंड डेवलपमेंट, जहां तक मुझे लगता है बहुत इंटरेस्टिंग टॉपिक है, फार्माकोलॉजी से रिलेटेड टॉपिक है, जो हमारे एमफॉर्म के स्टूडेंट्स हैं और फाइनल ईयर के स्टूडेंट्स हैं, बैचलर के, उनके लिए बहुत ही इंटरेस्टिंग रहेगा और कुछ ना कुछ इनको आज यहाँ से सीखने को मिलेगा। अगेन आई एम थैंक्सफुल टू डॉक्टर यशपाल एंड आई अगेन वेलकम टू ऑल द पार्टिसिपेंट्स इन दिस वेबिनार। थैंक यू वेरी मच।
Thank you, sir.
Thank you, sir, for your kind words. Now, I would like to invite the Registrar, IFTM University, Professor Sanjeev Agarwal, sir, to guide us and our students with his kind words. Over to you, sir.
Thanks, Harpreet. Honorable Vice Chancellor, Professor M.P. Pandey, eminent speaker, Yashpal Singh Chongar, and my dear colleagues, dear students. Actually, I am very much elated to be part of this webinar, role of designing and discovery and development. Actually, I express my gratitude to Dr. Yashpal Singh Chongar to give time to us from his valuable time, and I hope that this will be continued in future also. So the issue is very important. Pandemic gave us a challenge, and we accepted the challenge, and the entire world witnessed that we are combating with the pandemic, and the spirit we will be continuing. And this is not the end of the calamity. The calamities we will be facing in future, but our endeavor should be what? Should be to design and develop such a technique on which we can develop further techniques or further medicines we can discover, so that we can say that we are having cure, and now we are in search of diseases. Let the diseases come, and we are ready to combat and to cope up with them. So with these words, I express my gratitude again to Yashpal Singh Chongar, and expect that we all will be benefited by his valuable words, and I give my blessings to all. Thank you. Thanks, Arpit.
Thank you, sir.
Thank you, sir, for your valuable words. The main objective of organizing this webinar is to tell the pharmacy students that what is the role of clinical pharmacology in drug discovery and development. To guide our students, Dr. Yashpal Singh Chokar will share his thoughts and experiences. Dr. Chokar completed the M.Pharm degree from IFTM College of Pharmacy in 2007. Currently, Dr. Chokar is working as Research Assistant Professor, Department of Pharmacy Practice and Science, University of Nebraska Medical Center, Omaha, United States of America. Dr. Chokar peer research publication number is 70 and peer reviewed publication number is 5. The Google Scholar citation is 1394 with H index and I10 index of 19 and 38 respectively. These are just few of the career achievements of Dr. Chokar which I had shared. In this short period of time, it is impossible to share his works. Now, I would like to invite Dr. Chokar to deliver the talk on role of clinical pharmacology in drug discovery and development. Over to you, sir.
Okay. First of all, thank you very much for all of you for your kind words, and I'm always happy to connect my college. So IFTM is my journey, so I'm always want to connect to IFTM. So this is my luck to I'm presenting in front of you, all of you. So hopefully you guys enjoy, not get bored. So how can I share my screen?
So click on the present now.
Oh, I found, yeah. Yeah.
So your screen is visible now.
Okay.
It is visible.
So thank you very much again. And so today I'm going to present a role of clinical pharmacology in drug discovery and development. And so before going on the topics, I'm just giving like brief introduction, what is drug discovery and what is pharmacokinetic, what is clinical pharmacology. So if you like drug discovery is a collaborative programs, like it's not an individual person. So chemist design a candidates with good potency and activity, and the molecule go to biologist, and he will find out the mechanism action, and then candidate go to the pharmaceutical candidate optimization. Like it can be drug or not drug, because most of the molecule is not will be a successful a drug candidate. So then the journey start for the clinical pharmacology for safety, drug-drug interaction, dose projection, PKPD, and those things. And then the drug go to clinical discovery and development. So this is the drug discovery is a collaborative program. So clinical pharmacology role in the move the molecule from lab to clinical trials. And then further the back to lab and repurposing, and also identifying the liabilities and help to generate. So most of the compound do not become a medicine because due to the poor ADME properties and reason of drug failure, most of the drug failure in phase 1 to phase 3 clinical trial. So this is due to establishment now the clinical pharmacology now in the initial stage of drug development. So clinical pharmacology majorly is the role of the pharmacokinetics. As you know all about the definitions and those things, I'm not going in the detail what is the pharmacokinetics. So I'm just giving brief introduction. Pharmacokinetics means what the body does to the drugs, and pharmacodynamic is the what drug does to the body. So our role is to how the drug is moving inside body and how it is behaving, and our aim to how to drug is traveling inside body and how it is eliminating, because each drug is in our body is a foreign particle. So our body is behave like to remove as soon as possible. So those candidates are not like good ADME properties. It is failed in the later on the drug development. So in the pharmacokinetics, have you ever read the drug labels? Those all information come from the pharmacokinetics, like what should be the dose, how we can take, and what is the drug-drug interaction, what is precautions. Those all informations of the drug label, you will, when you purchase any drugs, there should be a leaflets describing the pharmacokinetic informations. So these all informations about the drug come from the pharmacokinetics or clinical pharmacology. So the general stages of so when we will take a drug, there is the 4 stages like absorption. First drug will be taken, and then it's go the systemic circulation, and there is bind to plasma proteins and go to reach to target, and then inactivation, metabolism, and excretion. So ADME, these are the four major steps of the drug when reach in systemic circulation. So initially, like when the initial drug development, we think that it's a linear process, and it's like take 15 to 20 years for a drug reach in the market. But nowadays it's not a linear process. Previously we will identify target, then lead identification, lead optimization, and then it's go for clinical candidate selection, that it can be a drug or not. So that is waste a lot of money and lot of time. So nowadays this is parallel process. At the time of the lead identification and we found a good candidate, we will start the parallelly the candidate selection. Like it can be a good drug, or it can be a successful drug candidate, or it's a poor activity, poor candidate for the drug selection. So in that way, saved a lot of time and money. So nowadays the drug discovery is not a linear process; it's parallel process. So if you see this two graphs, so before in 1960s to 1980s, 40% drug is failed due to the ADME or PK properties, due to these studies done in the later stage of the drug development, and so many drugs fail due to poor PK properties, like drug is not absorbable or it's not eliminating, because the drug is going inside body, it should be get out from the body. So these poor properties, the drug fails so many. But nowadays the clinical pharmacology involved in the initial stage of the drug development. So the failure of the drug is due to PK and ADM is only 5%. So what clinical pharmacology role in drug discovery? So when we will find a new chemical entity with drug-like properties, it's go to the pharmacology department for the pharmacology, and the same time it's go to the clinical pharmacology to find out the molecules is a favorable pharmacokinetic or it's a poor candidate to modify the structure or any modification required. So first we will identify what is the physiological properties, like what is solubility, chemical stability, microsomal stability. Then we will do the in vitro PK studies, like microsomal plasma protein bindings, and then if these are favorable, then we will go to in vivo PK/PD studies. And then molecule go for the IND application and clinical trials, and we will do the population. So because clinical trial initially will do the small amount of the population, and then using population PK, we will generate the how can we use that drug for the kids, how can we use the drugs for the male, females, and the region references. So this is the journey of the drug molecule in clinical pharmacology department. If those molecule required any modification, the molecule are not favorable for these all PK and ADME properties, molecule go back to lab and need to modification. So that make save a lot of time and money. So how we analyze the drug from the our biofluids like plasma or blood or tissues. So this is a separate topic, but I'm just giving brief introduction using bioanalysis, like using LCMS, HPLC, UV. We will analyze the drug in the is audible? My
Yes, sir,
voice is audible?
Yes, sir, you are audible.
Okay. So using these techniques, we will analyze the drug in different biofluids, and then we will how it's metabolized in the systemic circulation or in our body, and then we will correlate those concentration with the PD, like pharmacodynamic effects, and correlate these. And so just brief, we will make the validation parameter as per FDA, US FDA guideline, and then we will calculate the individual observation. Using those concentration, we will calculate PK parameter, like what is half-life, Tmax, Cmax, and then use the statistical result comparison and then relevant conclusion. So this is the how we will generate the PK parameter using bioanalysis. So our main aim is the lead identification and optimization. So this is the theoretical that the B.Pharm fourth year, you guys reading the Brahmankar. So whatever the all topics be in our lab, we will do all these experiment in the lab, like metabolism, pharmacokinetics, physiochemical absorption, transportation, modeling. So all these experiments we will do in our labs. So then we will using this data, we will use different software to generate the pharmacokinetic parameters, and then use non-MEM and adapt software for population PK. And then we will using PD data for the PK-PD correlations, and we will do the interspecies scaling, like using different species, like how the drug is behaving in different species, and then using the mouse data to predict the physiological based pharmacokinetics. So, and then molecule go to after this phase 1 to phase 4 clinical trials. So these are the few examples we did in our labs. Like when molecule come, first we will do the solubility, because solubility is the critical for drug molecule, because if the drug is not soluble in aqueous, that is very hard to win the formulation, and it will not absorb in our systemic circulation. The first thing is we will optimize the drug is soluble in aqueous or not. So first things we initially do these drug-like properties. If candidate fails, so this is two examples. Like one, this is not soluble, precipitate immediately when we dissolve in the PBS, even in 1 micromolar concentration, but another one is soluble. So, and same thing we will do the compound in vitro experiment, like compound is stable in a gastric or intestinal pH or physiological pH. If the compound is not stable, we are not able to develop that molecule as a tablet, because they are unstable in the gastric pH. Same thing we will do the plasma stability, just incubate the compound in the plasma and see it's stable or not. If it's unstable, we will discuss with the chemist and say back and need to modify the structure, or what is the poor site for the degradation of the molecules. So initially we will check these physiochemical properties for drug candidates. Then after this, the molecule is past those initial experiment, then we will do the in vitro metabolism. All these are the in vitro experiment we will do, like check the drug is metabolized by the liver enzymes, or it's stable in our systemic circulation. So these are the 3 example of 3 compounds we will in our labs. Like one molecule is immediately metabolized by the liver enzymes, phase 1 metabolized, and these 2 molecules are stable. And these are the another example of the prodrugs. Like this molecule is within 5 minutes 100% degraded and make another metabolite. So this molecule behave as a prodrug, and this is further metabolizing with 3 metabolites. Because every drug is in our systemic circulation either excrete as it is, but most of the drug is metabolized in polar metabolite and then excreted. So if the molecule is moderately metabolized or stable, then molecule go to further in vitro or in vivo PK studies. This is another example of another 2 drugs, the isomers. Those are the stables in the microsomes, and the molecule is metabolized by phase 2 metabolizing enzymes. So, and then this drug is within 5 minutes completely metabolized. Then we will find like how it is changing in the body and what is making and where it is going. So then we will found it is metabolized by the hydroxylation, and within 5 minutes it's giving as a 100% metabolite. So some drug like behaving as a prodrugs, they immediately convert to other metabolites like aspirin or so blockbuster drug. They also immediately convert in our body in the active metabolites. So this is the example. If the case like molecule even not stable in microsomal enzyme, but it's very active and very good activity, then we will find that there is any active metabolites or there is any other thing. So we found that these are the active metabolites, and we are developing this molecule as a prodrug. This is in advanced stage of the drug development, this molecule also. So after that, we will also do the interspecies scaling and see like because our ultimate goal for the any molecule should be developed in the humans. So we will do like mouse, rat, dog, and monkey, and all liver enzymes are available. So we will incubate the drug and see how the molecule behaving. So these molecules, because these are the all molecule in drug development stage, so I can't, they are not published, so I can't show their structure, but I just give their X and their name XYZ. So this molecule is unstable in monkey and mouse, but it's stable in human and rat and dog. So further studies for this molecule we will do in dog and rat, and on then scale up for the human. So these species also, like they are a species, but this molecule is not a species specific metabolism. So these things also initially do on the basis of the further experiment decide like what species is good for the further experiments. And after those all in vitro experiments and molecule is stable and its physiological properties are favorable, then we will do in vivo inject the molecule to the mouse or rat and see like how our aim to like monitor the drug in the over the time, like how the time over the time course the drug molecule present in our body. Or so these are the two molecules, though initially stable in microsomes. Then those we need to decide which isomer should we go further drug development. So RAM 2061 and RAM 2093 is very favorable properties in all and very good anti-cancer activity. And then our aim to decide which molecule, because we can't move both isomers, so we need to decide which isomers should go for further. So then we do pharmacokinetic studies and tissue distribution, and this we found like RAM 2061 is fast eliminating from the body, and RAM 2093 is like accumulating in and slow clearance. On the basis of this, we will figure out what is the tissue distribution, and we found that RAM 2093 is significantly accumulated in the liver, and that is also by PD results also correlate that this isomer is toxic in the longer way. So we will pick the RAM 2061 for the further drug development. So using these all experiment on the initial stage, we will decide like which molecule go for further drug development. And this we have recently published in Journal of Pharmacology and Clinical Experiment Therapeutics. And then we got the concentration using in vivo pharmacokinetic, and the pharmacologist get the pharmacodynamic result, and then we do PKPD and decide the dose and those what is the next dose things using those correlations. So is these all things we will get like all preclinical experiments, and using this preclinical experiment we will not go directly for the clinical trials. We will then nowadays do the simulation like physiological based pharmacokinetic modeling. These are the all mathematical modeling, and nowadays these are for any drug approval, those things like required, like using mouse or rat data. We will generate the human data by using mathematical modeling, and what will be the human first dose, and what is the how can we repurpose any drugs. So using physiological based pharmacokinetic, we will do these things. So this is the one of our example. We Genfinitive, we have mouse preclinical data, and then using this modeling, we will generate all human data, like what will be the plasma concentration in human and his tissues. On the basis of these all experiment, we will decide what will be the dose, first dose in humans. And this is another example of physiological based pharmacokinetic and scale up to humans. So these help us to reduce the times and a lot of money. These nowadays, like using preclinical data, we can generate human data, and after if these this look like favorable, then the molecule go for the clinical trials. And the clinical trial, as you know, in 4 phases. So phase 1 is generally for the what will be the first human dose, and what is the maximum tolerated dose, and then PKPD and toxicity. And this is the doing a small amount of the volunteers. And then phase 2nd to phase 3rd is generally used for the PKPD and relation, toxicity, efficacy, what is food effect, it should be taken with empty or it with food, and what is the drug-drug interaction, what drugs should be avoided and what's drug not avoided. So these all is what and what how that drug in the pediatric and renal impairment or pregnant women. So these all parameter we will cover in phase 2nd to phase 3rd. And then we repurposing and address the liabilities and IP, intellectual property rights, in these. And then 4th, if these drug pass in these, then they go to phase 4 clinical trials. So when the molecule we will do clinical trials, then we will also, so clinical trials generally do a small amount of the volunteers. So then using that data, we will do the population pharmacokinetics. That also another mathematical modeling using softwares, and we will generate like what is the how the drug will be behave in the special population, if any renal impairment or pregnant women or those things. So without doing those experiments, we will generate all that simulation and do the what is the parameters that we need to take care. So this is one of the example we did in the population pharmacokinetics in the testosterone in healthy male subject, and figure out how it will be beneficial for the other populations. So this is the whole drug discovery story. Like we have a pharmacological effect, and then we will do in vitro pharmacokinetics, and then we will tissue distribution, and on the basis of this, like how drug is accumulating or in the tissues, and on the basis of these all tissue distribution, we will select molecule, and then we will do PKPD correlation using the pharmacodynamic effect and our PK concentration. We will make correlation and decide what will be dose. It should be given single dose in a day or multiple dose. So basis of the concentration and minimum required concentration, we will select all those. And using all this in preclinical data, we will do physiological based pharmacokinetic and generate human data. That data will be helpful for what will be the first human dose for the clinical trials. And then we will do the clinical trial and then molecule. After clinical trial, we will do population PK for the special population, or because we can't do the clinical trial in each and every population. So using the initial clinical trial data, we will generate data for the other populations. And if we found like anything serious, we need to take care of that, then the further studies go on for that things. If there is no effect, like if that same drug can be given for male, female, or children's, or elder old persons, or if not, we will take care of that, like need to dose adjustment or dose reduction or anything. So we will take care for that using population PK studies. So this is the all steps we will use in clinical pharmacology departments to how the molecule move forward and back from clinic to lab to repurposing. So nowadays we also working 2-3 clinical trials for the repurposing for the hydroxychloroquine for COVID and ivermectins. So these are the repurposing, like if drug have some other activity but need to address another activity that called repurposing, and nowadays those things also we are doing in the lab. So for that also, like, we will correlate how it's affecting and concentration correlation with pharmacodynamics. And this is another thing, like we will also do the pathway analysis, like when we any drug molecule, as because every drug molecule is foreign particle for our body, and when it's reach in our systemic circulation, it do changes in our biochemical metabolites. So in our body there is lot of metabolites, endogenous component like amino acid, bile acids, thousands of chemicals are there. So those we call biomarkers, like and they are variable and characterize cellular organs and clinical conditions. So using these like how they are changing when we are giving treatments, and find out the what is the pathway is upregulating, downregulating any specific. So in disease condition, if any metabolites or any biomarkers upregulated or downregulated after treatment is coming in the normal condition or not. So we will using this metabolomics analysis, we will analyze all these metabolites using LCMS and inflammatory biomarkers, short chain fatty acids, and see like how they are changing in the disease condition or in the after treatment. So this is some example of the healthy versus cancer patients. Like most of the metabolites like glutamate, they are increased in the cancer patients, but in the when and they are very distinguished endogenous metabolites. And then we will go for the treatments and then see like those metabolites come in the normal or not. And on the basis of these, we will find out what is the pathway of the is the drug is upregulating any pathway or downregulating any pathway, specific pathway, and on that pathway is affecting any other thing. So we also correlated these pathway analysis and metabolomics using these endogenous metabolite analysis. So on the conclusion, I'll say the clinical pharmacology tool is facilitated and identification, optimization of the lead compounds, and using the concentration versus PD effect correlation. And then we decide dosing strategy from animal to IND application, like what will be dose in the animals and what will be dose for the IND application, what will be the dose for the first human dose. And then we will design clinical trials, phase 1 to phase 4 studies, and then principle applied to across disease state and therapeutic areas. And that is these all experiment is reduce the animal experimentation and also reduce the time. And if the molecule is poor in the those all ADME properties, we will back to send the molecule to chemistry lab and modify those things, and that make the lot of times and money. And then strategies individualizing drug therapy in clinical using PK, PD, and PG pathway analysis, and that is risk reduction for drug development. So our main aim is to reducing the risk for the if it's going molecule in clinical trial and then its fail is very expensive and time consuming. And so our role is reducing the risk for the drug development failure in the later stage. And that's our role as a clinical pharmacologist to develop in the drug discovery. So these are our lab members, the lab director, Dr. DJ Murray, I acknowledge them, and these are our PhD students and our alumni and all these collaborators. I'm thankful for all of those projects, and these are our fundings. All projects are funded by these all national agencies, and that's it. And this is our first batch of the IFTM. We are in 2005 to 2007 in the year, and always good memories with IFTM. So I'm very thankful to you guys. Give me opportunity to talk to you, and if you have any questions, you can ask.
Thank you so much, sir.
Thank you, sir.
Yeah, and about expect this. So you all are in B Farm 4, so I'm not able to see the videos.
What? Sir, your voice is audible. Please continue.
Okay, so this is my research. So if you guys have any questions regarding, so from our, so I'm belong from Rajasthan, and I study in the UP. So from our this region, the very less person come to in the United States for the PhD and those things. So if you are in the, so if you are planning to do GRE and after B.Pharm final year, if you have GRE score, you can directly join the PhD program here. So if you have any question regarding for the GRE, TOEFL, and how to get admissions, you can reach out me anytime and take my emails and email me directly of any question regarding for how to future directions of, and any help I can do. I'm happy to help my colleagues, and I'm very thankful for all of you giving me this opportunity and very kind word for introductions. So I'm very thankful for all of you.
Thanks, sir, for sharing the in-depth knowledge with us and our students. Sir, students wish to know the parameters which must be taken care while drug development for pandemic diseases like vaccine development for COVID-19. So please elaborate.
So we are basically, we are not working on vaccine, but we are basically working on anti-cancer, anti-HIV drugs. So we are working on 2-3 clinical trials of COVID for repurposing drugs like hydroxychloroquine and azithromycins, but those are failed. But initially, those are very famous for the COVID initial treatment. But those we have analyzed like how it's working and what is their mechanism action. So, but vaccine is not our expertise area.
Okay, sir, thank you. Thank you, sir, for
Yes.
sharing your knowledge. Students, the feedback link will be provided in the chat option and to your WhatsApp groups. After filling the feedback
Okay, sir.
link, you will get your
Okay, sir.
e-certificate. Meanwhile,
Okay, sir. Okay.
I would like to welcome the organizing secretary, Professor Sushil Kumar, sir, for vote of thanks. Over to you, sir.
Thank you, Harpesh ji. My loud is
Yes, sir,
you
you
are
are audible.
hearing?
Please continue.
Okay. Thank you, Dr. Yashpal Singh. Firstly, I am very grateful to you to interact with our students as alumni. Really, I am very happy. And also, since a long time, I am interactive today. Really, it is a very grateful moment for all of us. And thank you, Yashpal ji. You have shared so many things regarding the role of clinical pharmacology in the drug discovery with our students. Definitely, this presentation helpful for the research students, either those who are interested in the PhD programs, yeah, they are involving in the research program. Definitely, these things very helpful to our students. I am thankful to you for your sharing so many things regarding the pharmacokinetics, pharmacodynamic, these parameters, how we can calculate during the discovery programs. Really, I am very thankful to you for your support with our students. Thank you, Yashpal.
Thank you, sir. Thank you. Thank you very much, sir.
I am thankful to our Honorable Vice Chancellor, Professor Mahendra Prasad Pandey, sir, for their motivation and support to conduct this webinars. I am also thankful to respected Registrar Sir, Professor Sanjay Agrawal Sir, for sharing his views regarding the role of clinical pharmacology in drug discovery. I am thank you, sir. I am also thankful to Alumni Association President Professor Anush Srivastava Sir for their supports. I am also thankful to Dean, Faculty of Pharmacy, Professor Navneet Verma Sir, for their support and interact with our students and sharing his views during the webinars. I am also thankful to Professor Arun Kumar Mishra and Dr. Kavita Gehlot for their support and their motivation to conduct these webinars on the role of clinical pharmacology in drug discoveries. I am thankful to Dr. Harpreet Singh for their supports, and really he is taking full support regarding the conduct of these webinars. Thank you, Harpreet, for your
Thank you,
team,
sir.
and also to you to conduct these webinars. Definitely, this webinar helpful to our students regarding the higher studies. So I am thankful to all the organizing teams and faculty member and staff and their students for participating in these webinars. So thank you to all.
Thank you, sir, for your kind words. Now, students, furthermore question you want to ask? Any questions from students?
No, sir.
Okay.
Sir,
Thank
one.
you, Yashpal sir, for giving.
Harpreet sir.
Yes, sir.
One of us.
I'm the expert of the pharmacokinetics. I will basically work on the how the drug is moving inside body, and there is a lot of models are there. So those our collaborative pharmacology person do that things, and we basically do the all ADME, absorption distribution, pharmacokinetic, clinical trials. Those things are we are basically involved in these things.
Hello.
Thank you, sir.
Hello.
Yes, Shurvi Srivastava.
Hello.
जी
Yes.
मैम पूछिए.
Good morning, sir. Sir, I want to ask that how can we take admission for PhD if we want to do from USA or some good institution
So,
out of India?
so here like a lot of student from so in our university, lot of student from the Bangalore and South India, they just do the preparation for the GRE and TOEFL. If you got the good score and the GRE TOEFL, and you can directly apply for the different institutions through online, and they will you will selected in the university. So you need to apply in different institutions after getting the score in the GRE. If you need the information, our recently 2-3 students join our from India recently from Mumbai and those, and I can share their contact number and they can help you to how to do preparation, because after
Yes.
PhD you can come directly, but if you are after coming masters or B forms, you need to qualify those GRE and TOEFL, and then I think those are not very tough exams. So if you guys want those tests, I can share some information. I will
Yes,
connect
sir,
those
definitely.
join, and they can help you providing the materials and those things. So if you interested, I can share those contact numbers to you.
Yes, sir, sure. I'm interested in this.
Yeah, sure.
Thank you, Yashpal sir, for giving us your valuable time. Now the webinar is over. If the students you want to leave, then you can leave.
Okay, sir. Okay.
Thank you very much,
Thank you so
everyone,
much.
again.
Thank you so
Thank you,
much,
sir.
sir.
Thank
Okay,
you,
sir.
thank you.
Thank you, sir.
हमने लेफ्ट कर दिया